Dr. Sonia Shah, National Heart Foundation Future Leader from IMB and University of Queensland. Sonia's research leverages genomic data to better understand human diseases, with goal to improving prevention, diagnosis, and treatment of cardiovascular diseases. Sonia is going to present her data on development of clinical application of large-scale genomic data.
Thank you so much to the organizers for inviting me to speak today. So I head the Genomics in Health group at the IMB at UQ, and my research program really has 3 arms to it, but I'm going to focus on the cardiovascular genetics. And I hope to take you through a very fast-track journey from the birth of genome-wide association studies to really where we are today in terms of translating those findings. So the field of genetics has been very successful in identifying the genetic basis of many human diseases through linkage analysis of large families or pedigrees. But one of the reasons for the success is because of the underlying genetic architecture of these diseases. These are generally rare disorders, are monogenic in that there's a single genetic variant that has a large impact on the phenotype. And so through these smaller family studies, we have the power to detect these variants. However, applying the same approach to more complex common disorders really hadn't been very successful. And we do know now, thanks to over 15 years of genome-wide association studies, that again, that came down to the fact that complex disorders have a very different genetic architecture. So in 1996, this paper came out in Science by Rish and Marikangas, and they suggested that one of the reasons for the lack of success was the fact that these genetic variants maybe only have a moderate effect on complex disorders. So we really didn't have the power to detect them. And they proposed that a genetic scan using 1 million variants in unrelated individuals could actually be more powerful than doing linkage analysis with a few hundred markers. And they ended this paper with a statement that it is a charge to molecular technologies to develop the tools to meet this challenge. And indeed, since the completion of the Human Genome Project and the subsequent development of genomic technologies, this became a reality 10 years later. So really, GWAS studies or genome-wide association studies were designed to, one of the beauties of it is you can do these experiments without knowing anything a priori about the disease. And so it had a lot of potential for identifying novel disease biology and potentially novel drug targets. But one of the things we really realized from doing such studies is that common complex diseases have a polygenic architecture. So it's not a single genetic variant, but multiple genetic variants across the genome that contribute to disease risk. But what we also learned is that I should have explained what this is. This is a Manhattan plot for those of you not familiar. This is how we visualize genome-wide association studies. Each dot there is a genetic variant in the genome, and the peaks, so if the bigger the peak, the more significant the association between that genetic variant and disease status. And so it's called a Manhattan plot because it should look like the Manhattan skyline. But one of the things we learned that the effects of these variants that we were identifying from GWAS actually had very small effect individually on disease risk. And so this paper that came out in 2010, there was a lot of skepticism about whether GWAS could, or is identifying any relevant disease biology and could have any clinical application. And I hope by the end of this talk, I can convince you that maybe this statement was made too early, and indeed there is clinical utility. So yes, these genetic variants had very small effects on disease risk individually, but what we know now is that the collective effect or the cumulative effect of these risk variants can actually be much more sort of clinically relevant. So each of us, we all carry a unique set of these genetic risk variants for every disease. But if we have a very large number of these risk variants, our genetic predisposition to disease is much, much higher. And this is where polygenic risk scores came about. It essentially provides a single estimate of an individual's genetic liability to developing a particular disease. But again, this is going back to 2010 with that paper. And as I said, there's a lot of skepticism whether this could have any clinical utility. And this is work that was done during my PhD. And here in this study, we actually showed that a polygenic risk score for different lipid fractions could actually predict future health events or outcomes. And over here, what we're showing is different polygenic risk scores for the different lipid fractions were associated with future use of lipid medication, but also not in this particular plot, but we also show there's an association with developing coronary heart disease as well. And I was sort of happy to learn that Professor Sir Michael Marmot is actually going to be here tomorrow, but this was work done in the Whitehall 2 study, and I believe he was the first lead investigator of that study. In 2018, this paper came out from Mike Inoue et al. In JACC. And here what they did was they analyzed data in over 300,000 individuals in the UK Biobank data. And what they showed that a polygenic risk score for coronary artery disease on its own actually was a better predictor than many of the established risk factors for cardiovascular disease. So you've got things like smoking, diabetes, family history of heart disease, BMI, hypertension, but actually the blue dot here is the polygenic risk score for coronary artery disease. Of course, if you add all the traditional risk factors, you get a much better predictor of disease, but then integrating polygenic risk score into that further improves prediction of disease. One of the interesting things that came about was PRS is actually independent of family history. And that's probably because family history is capturing more monogenic variants and also shared environmental factors. But one of the things about polygenic risk score is it doesn't change over time. You have that genetic risk from birth, which really provides an opportunity for earlier intervention. And we know now from work from Gemma Figtree that there's an increase in the number of people who are having a heart attack without any traditional risk factors. And so potentially polygenic risk scores could be useful there. And also a nice thing about it is we don't actually need to know the underlying biology of these genetic risk variants. We can still use it for prediction. And this is already starting to be done worldwide. So in the UK, Genomics PLC is a company, and they've done the world's first pilot of using a cardiovascular risk prediction tool which integrates polygenic risk score into the NHS. And what they're reporting based on preliminary results is when you use the polygenic risk score, 5% of their study sample was actually reclassified to high risk. So they became eligible for intervention. And also GPs report that they would change the management of around 13% of the overall study population. And based on modeling, they suggest that using an integrated risk tool could actually cut the number of cardiovascular deaths in England by 11,000 over 10 years. So this is an example of how going just from disease biology discovery, how it could actually be used in disease prediction. But this is another application that it probably wasn't something that, or sort of, I guess, came about unexpectedly, was that it could potentially be useful in the context of monogenic disorders as well. So familial hypercholesterolemia, as many of you know, there are sort of three genes where we know that there are pathogenic variants within these genes that can cause FH, and genetic screening programs really look for pathogenic variants within these genes. But in around 60% of patients, no pathogenic variants are found. And there is incomplete penetrance in variant carriers as well. And in this study that I was involved in, we actually looked at whether a polygenic risk score for LDL cholesterol could actually explain a clinical diagnosis of FH. So in this study, we had a cohort of FH patients who had undergone your traditional genetic screening, and we looked at their polygenic risk score for LDL cholesterol. And what we found that in a large proportion of patients where no monogenic variant could be identified, they actually sat in the top end of the polygenic risk score distribution. And our results have been sort of validated in independent studies. So this one that came out in 2018, they actually showed that people who sit in the top end of that polygenic risk score distribution, their LDL cholesterol levels are as high as people who carry a monogenic variant. But we only screen for these monogenic variants who make up only 0.3% of the population, whereas these people make up 5% of the population. And just quickly, again, more work that's shown that even within variant carriers, so these top three groups, the underlying polygenic background of these individuals can actually be used to stratify people as well. So in the UK, polygenic risk scores are already being used in some lipid clinics in the context of FH management, and really to sort of in people who have both monogenic and high PRS, they might require more intensive treatment, whereas people where they only have a polygenic contribution to their clinical diagnosis, it sort of removes the need for cascade screening because the risk to their family members is not as high. So there's potential cost saving as well. And a nice thing that is that clinicians who are using this polygenic risk score actually reporting that knowing a genetic cause for the disease, even if it's polygenic risk score, actually gives them more clarity about their diagnosis and less guilt that their high cholesterol is due to their own actions. And they're so more likely to accept treatment as well. So in terms of risk prediction, but also in informing disease management, and one thing I haven't talked about is genetic epidemiology, which we do a lot of, and it really helps us understand causal relationships between risk factors and disease. But I just want to finish off, given the theme of the meeting today, with this diagram, and this is figure that came out in Nature Medicine a couple of years ago. And what this is really showing is the participants of these genome-wide association studies and their ancestral, or their sort of ancestry breakdown of these participants. And as you can see, that the majority of participants are of European ancestry. So why does that matter? Well, if we're developing polygenic risk score in a European population, when we go and apply that to other populations, we know that they're not as accurate. And so we're sort of going ahead in trialing these within the healthcare system, but we know that there's already bias in how well these perform in other populations. And this is just something that we published on recently in the Australian Quarterly in the context of cardiovascular disease. But I just want to say that there are efforts internationally and in Australia to try and address this diversity gap. You have the National Indigenous Genomics Network, our DNA study led by Dan MacArthur, but also the South Asian Genes and Health in Australia, which I'm the lead PI on and is funded by the MRFF Genomics Mission. But really, these are studies that aim to engage those underrepresented groups to ensure they're included within these sorts of research. And really to ensure that the benefits or the translation of genomics research happens in an equitable manner, we need to start addressing this gap now. Thank you.
Just touching on your last point there about the diversity issue for risk prediction, I guess you're implying then that if the genetic architecture differs between populations with regards to the disease, are we thinking of different diseases in different populations and potentially different treatments as well? Is that where that goes?
So the genetic architecture is not different across different ancestries. Yeah. So it's likely that it's just we're not, because of the lack of representation, we're not accurately estimating the effect of those risk variants in those individuals, and that's where the sort of lack of accuracy comes. But the genetic architecture is the same, but there will be sort of less common variants that are very specific to certain populations, which we're not capturing in these scores. So it's really about ensuring that we're doing these genome-wide associations in these different populations, which have the same power as we currently do for European populations.